Good morning, good afternoon, and good evening, everybody. Thank you for joining our webinar. My name is Mehmet Tulbek. I'm the president of the Saskatchewan Food Industry Development Center. Today, we'll be hearing from Dr. Chris Eskew about the science and innovation behind Canada's microbrewing industry. And before we start, I'd like to go through a quick couple of slides about what Food Center does and our ecosystem and how we support companies from startups to SMEs and all the international, national and international companies. Food Center is a contract research and a contract manufacturing organization. We've been working since 1997 within the agri-food industry and the agri-food ecosystem, based in Saskatoon and supporting primarily on local crop, meat utilization, plant-based foods, ingredients, and within the agri-food transformation system. We have over 60 full-time employees with diverse expertise and specialized skills. And the last 20 years, we supported over 520 clients and produced and commercialized over 1,100 products. Our main goal is to expand and drive the innovation with Saskatchewan inputs to develop processed agri-food products. We support entrepreneurs, provide market access, proof of concept, provide reduced capital risk for entrepreneurs. And we also develop the ecosystem, commercialization of new technologies, research development, and in particular on the co-product utilization, as well as within the new ingredients coming into the marketplace. We are part of the whole in Saskatchewan agri-food processing and innovation ecosystem, but we are global clients. We work with several clients around Canada, United States, Mexico, and other parts of the world. So we have 3 facilities, our pilot plant in campus, about 10,000 square feet, our Agri-Food Innovation Center here at the Schuler Street, about 57,000 square feet, and most recent Advanced Food Ingredients Center at 8,000 square feet, primarily focusing on fermentation, solid state submerged fermentation with the downstream processing and drying capabilities. So with all those capabilities, we support the industry. With that, I'd like to introduce Dr. Chris Eskew. And Dr. Chris Eskew is a faculty in the Department of Food and Bioproduct Sciences and the Department of Biochemistry at the University of Saskatchewan. Dr. Eskew completed his master's degree in anatomy and cell biology at the University of Saskatchewan and achieved his PhD at the Institute for Medical Science at the University of Toronto. Dr. Eskew continued his research by moving to the United Kingdom for postdoctoral fellowships at both at the University of Oxford and University of Cambridge. Dr. Eskew was faculty at Brunel University in London from 2010 to 2013 before returning back to Saskatchewan. And he established his lab in nutrigenomics and genome organization at the University of Saskatchewan. Dr. Eskew has since applied his knowledge of genomics into yeast, establishing a research program focusing on the genetic difference between specific brewer strains and how these differences contribute to different beer flavor profiles. So in today's webinar, Dr. Eskew will provide an overview of the microbrewing industry in Canada and discuss some of the underlying scientific principles, the challenges likely to be encountered, and the trends that would dictate the continued success of the microbreweries. With that, thank you, Dr. Eskew, and I'm leaving the screen to you.
Thank you very much, Mehmet, and thanks everybody from all over the place who's attending today. Really appreciate your engagement. I guess I should start actually at the beginning of my talk here, so my apologies. It's always some sort of technical challenge, no matter how prepared you are. So again, thanks for showing up today. Really appreciate it. So I do want to talk a little bit about the research going on in my lab, as well as some of the product development I'm doing in my company. I'm going to focus on the science. I don't really want to talk about my company, but given the knowledge that we generated from the research, I have started a little side business providing yeast for craft brewers and distillers, and the company's called IntelliYeast Laboratories. So first things first, I love beer. I have loved beer for a long time. I really developed a taste for beer when I lived in the UK. Got exposed to a lot of European and import beers. In 2019, my wife and I, Nicole, were fortunate enough to attend Oktoberfest in Munich. And wow, just the culture around this, the party, the tent that we're in, there's 5,000 people. They're all dancing on tables, but everybody's really well behaved. You don't want to misbehave at Oktoberfest, but this was the best party I've ever been to. Not only do I like beer, I think my beer likes me too. I wish this was my picture, but it was pretty cool to include this. But why are beers and wine and spirits and other alcoholic beverages so important to us? I want to go into a little bit of background behind this. Beer is not something new. Fermentation has been actually predicted to be a driver of civilization as early as potentially 10,000 BC. There's a histogeographical region in the Eastern Mediterranean where there is evidence of early brewing and fermentation that had been occurring. And I'm not going to try to pronounce the name of this culture, but again, Eastern Mediterranean in its origins. But some recent research from Simon Fraser University is predicting that the transition from hunter-gatherer cultures was actually driven by the production of barley and seeding, growing crops of barley for beer production even before bread. So this is a pretty important aspect of human civilization and our development. Certainly, there is evidence of brewing from the Egyptian culture around 3500 BCE. There's evidence of breweries and brewing equipment. Even in one of the Egyptian tombs, there was an ancient urn or vat that was found to contain yeast that was buried and interned with one of the pharaohs. And there was a company in Egypt that had taken that yeast and was making a commercial beverage with it for a while. It was apparently a very odd, it's not in production anymore, but certainly you could have got it and that yeast was still working after almost 5,000 years being inside of a tomb. But again, this is a major driver of our civilization. Even some predictions that the domestication of maize in Mexico was for fermentation purposes. So yeah, all over the world in separate instances, we have fermentation and beer production driving early civilizations. So pretty cool. Here I have a picture of the god Hathor, and one of the myths, Egyptian myths behind this is the goddess Hathor was going to destroy humanity, but to stop this, one of the other gods got her drunk, and then she decided not to do it. So Hathor is associated with the goddess of drunkenness, but again, these traditions of beer are heavily rooted in human civilization. So, what are some of the other benefits that actually provided good things to the ancients? Well, certainly socialization. I don't want to talk too much about the psychology of food and feasting, but we do know that getting together, sharing food, sharing drink is an important part of our development as a society. So, again, beer is going to play a major role in that celebrations, revelry. Sanitization is also really important. When you're making beer, you boil the water, you do this mashing stage, which kills a lot of pathogens. And even though the ancients may not have realized what they were doing, this was beneficial for killing off those pathogens and making water sources safer to drink. Certainly preservation, when you have fermentation, this is lowering pH, consumes residual sugars. So if you have yeast that is growing in there, it consumes those sugars so that other harmful pathogens may not take hold in those solutions and cause disease, but also produces ethanol, which is going to suppress pathogens as well. Also, with some of the ancients, and I think this was quite prevalent in England and Scotland, beer was used as a nutrient source. Certainly in Edinburgh, Scotland, there's tales of prisoners receiving half pints of stout a day as a nutrient source because there wasn't enough food to actually feed them. They were given a little bit of beer that was rich in iron and the other helpful components of the beer as part of their daily uptake. And certainly throughout the Middle Ages, the monks were brewing beer to clean up contaminations in the water. And this is well documented with the Trappist breweries in Belgium and France, that the monks were doing this to purify or clean the water. And they have some modern technologies that I don't have a chance to talk about here, how the monks are actually using their wastewater and purifying it. They've got some really crazy, cool ideas just from that setting. And certainly, again, I have Oktoberfest up here. Again, that socialization aspect that is still ongoing today is really important for our culture. Okay. All right, well, let's talk about Canada now. Let's move into modern times. So I jumped ahead to a slide. Well, when we look at the amount of breweries in Canada, we have about 1,200 breweries. And this was an increasing value, especially through the pandemic. There was a lot of breweries that started during that time. Craft brewing, microbreweries are really a trend that has been heading in an upward direction over the last 10 years. And this definitely expanded over the pandemic. During the pandemic, there was a surprising increase in the consumption of craft beer. People were at home, they were a little bit bored, they wanted to try new things. And this really drove the production of new beers and new beer styles. So again, about 1,200 breweries in Canada. Some of my statistics are a couple of years old here, but we'll stick with it for now. 94% of the breweries in Canada are craft. And what we define as craft are those facilities that are producing less than 1.5 million liters per year, or less than 15,000 hectoliters, for those of you who are familiar with the jargon. In Canada, we love our beer. We love our domestic beer. And about 85% of the beer purchased in this time frame is from Canada. We do bring in some imports, some European beers, some American beers, but again, we do drink mostly our own beer. It is predicted that there's greater than 10 million beer drinkers in the country. There might be a start towards a decreasing trend here, and I will talk about that a little bit later on. Post-COVID impacts and also health trends, but I'll talk about that a little bit later. When we look at the distribution of how this beer is actually being sold, 74% of that is actually distributed in cans, 21% is in bottles, and the remaining 5% is in kegs. And this is likely for the restaurant industry itself. So getting those large volumes in that way. But again, people are buying a lot of beer to take home, take camping, vacations. This is the primary form in which the beer is sold. All right. Well, what about the distribution of craft breweries throughout Canada? There is certainly some hotspots. BC, Ontario, Quebec basically have the lion's share of the craft brewing numbers as far as breweries are concerned. Here in Saskatchewan, there's 40 craft breweries that do excellent work. I'm a big fan of the breweries here in Saskatchewan. I actually never met a brewery I don't like, let's face it. I love beer. I love the different artistry that's going on. One small point about these numbers that we have to take into consideration is for any business that has off sales in their beer, they have to make a small amount of beer to get that off sales license. And they may not be bona fide craft brewers. They may do this just so they can sell an off sale. So these numbers might be a little bit misleading. The number of what I'd call real craft breweries here in Saskatchewan is probably about 28, 29 of this 40. So I don't have the statistics on the rest of the provinces, but yeah, it is nice to see in Eastern Canada there are a fair amount of craft breweries there. There's a lot of traditions there, and they make some great beer. So yeah, we're doing this countrywide, and I think we're doing a fantastic job with this. The economic impact of the beer industry. This is really quite significant and maybe something that goes unnoticed. Approximately 1 in every 120 jobs is directly or indirectly involved with supporting the brewing industry. And given how much farming we do in barley production, this is not surprising. In 2016, $13.6 billion from the brewing industry contributed to Canada's GDP. So this is, again, quite a significant impact. Unfortunately or fortunately, depending on how you view taxes, Canada has one of the highest taxes on alcohol and beer production in the world. And this generates about $5.7 billion for the country. If the taxes get used for the right thing, again, that's a political discussion. This is a good thing. It generates a lot for the country. And there are approximately more than 149,000 jobs associated with the brewing industry, resulting in $5.3 billion in income for folks. In Canada, for barley production, greater than 300 tons of malted barley is produced in the country per year. And I do have some numbers here. I don't want to talk too much about it. There's 2-row and 6-row barley. There's a lot of new varieties of barley that are being produced for the brewing industry. And that's where I'll leave this. This is a really important aspect of Canadian agriculture. And here in Saskatchewan, we're developing these new varieties that are optimized for malted barley production and the brewing industry. But that's another talk for another day. But certainly we can't have beer without barley. It's an important aspect, an important part of our economy. So let's get down to brass tacks here. What is beer actually? It's simple, and I'm going to say that, and then I'm going to put that in quotation marks. Beer is fairly simple. It is a combination of malted barley, hops, water, and yeast. You mix it together, and magically you get beer. It sounds simple. I do want to bring up the Reinheitsgebot. If for anybody who speaks German out there, I'm sorry I just butchered that. But these are the beer purity laws that were introduced in the times of the Holy Roman Empire that said that if you were going to make something called beer, it could only be a mixture of water, hops, and barley that could be added together. The yeast at that time obviously wasn't known, but it's an important component. But to make beer in Europe at this time, you could only have these 3 ingredients. If you added anything else, you couldn't call it a beer. So again, it sounds really simple that we are just mixing these components together and magically we get the diverse flavors, the ethanol, and the purification of water that we all love so much, right? Sounds simple, but really what is causing that diversity of flavors and the different types of beers that we see? I have some screenshots here of different beers you can find on the shelf. I'm hoping that whoever took this picture took this in a 4-degree fridge because beer sitting at room temperature on a shelf is not a good idea. This is a good way to take a good beer and make it bad really fast is by leaving it at room temperature. Another story for another day. But this diversity of beer flavors that we love, where does it come from? Well, the recipe is really important. Getting the right mixture of malt, which is our germinated barley grains, toasting and roasting those grains to varying degrees to generate different flavors. And also you can add a little bit of rye or wheat malt in there as well. So that's really important, absolutely. And these malts provide the sugars and the flavors such as chocolate, coffee, raisin, etc. to the recipe. The hops also is contributing to the flavor profiles. There's a ton of different hops. I'm not sure how many varieties there are now, but they're constantly in development. And these are adding potentially citrus flavors, bitterness, or pine flavors, for example. So getting this recipe right is really important. But the key driver to flavors inside of a beer is the yeast. The yeast is, for each beer style, you have a genetically unique yeast that is added for that particular style. And the yeast is taking those starting materials and converting them into ethanol and the flavors that we get in each one of these styles. So the yeast is really the driver for this. They are the biological factories that are converting those starting materials into volatile or flavor compounds, as well as the alcohol. I do want to bring up this term here called systems biology. And this is the study of biological systems, as the name suggests, in which you take into account the totality of the system that you're looking at. And again, so we're talking about beer. You throw it in a container, it has those four components. Simple, right? But you need to take into account the minerals, the nutrients that the yeast have. The temperature is really important. The pH is really important, but also the size and the shape of the vessel you are doing this in. If you have something with corners, for example, if you don't have a nice rounded vessel, the yeast gets stuck in the corners and they get stressed out and they start to produce a bunch of off flavors that really don't go well in a beer. So to contemplate the entire system, this becomes, even with just these four ingredients, it becomes a really complicated system that really needs care and attention to detail to devise what's actually going on. So I just have some pictures of some yeast here. This is a dark field image here. Yeast look really simple. Here's an scanning electron micrograph of some yeast. And every time you see one of these rings, that's a scar from where this yeast has produced a daughter cell. It's called a bud scar. And then we just have some bright field microscopy. So they look really simple. And they are, as far as uh, eukaryotes or organisms with a nucleus. When we consider yeast, they are very, very simple, but they still do a lot of complicated things. And like I said, each one of these strains for each brewing style is genetically unique. So let's talk a little bit about the genetics of these different yeast strains. So in 2016, Brigitte Glone from the Verstrepen lab in Belgium, they took 157 different strains of yeast and they sequenced their entire genomes to find out why these yeast were different. Why are they genetically unique? Now, what they did when they did this whole genome analysis was they looked at how the yeast had evolved over time using something called phylogenetics. If you don't know what that is, don't worry about it. It is just a computational method to look at how evolution has occurred. And what the Galoni colleagues had identified is that when you look at beers from different geographical regions, they evolved together. You can group them. For example, let's take the beers from the United States here. They branched off from the beers from Belgium and Germany right around the time that the New World was beginning to be populated. So a yeast likely made the jump with the people coming over. And as the brewing conditions changed to the new environment, these yeast started to evolve over couple hundred years to produce new varieties that were very specific for that geographical region. In Britain, again, this branched off from the United States strains, but again, all the British ale strains of yeast clustered together, showing that they're evolving in a very similar way. And this is likely due to environmental conditions, such as the temperature of the brews that were being made, the ingredients that were being used, the type of malt that was being used, is forcing the evolution and the change in these yeast in a specific direction. When we look at the wine strains, you can tell that these all evolved very differently than the beer strains, and they are very genetically unique. You certainly don't want to take a wine yeast and try to make a beer with it, or vice versa. They make terrible products. But it makes sense that they evolved in very similar ways for what they're doing. But certainly in this population, when we look at the geographical regions, the geography is what it was dictating that evolution. Here, if for you science nerds out there who understand principal component analysis, when they looked at the principal components of these genomes, they certainly grouped together, basically confirming that the evolution of these yeast was happening because of the geographical region and likely the starting materials and the style of brewing that was going on in those different geographical regions, which is pretty cool to see. Unfortunately, with this paper, the authors called them beer 1, beer 2, beer 3, and didn't tell us what type of beer they would actually make. So if you tried to go out and purchase a yeast because you're like, oh, this one looked really interesting, what is it? The authors will not tell you. And that's disappointing. I've asked them for this data several times, but Nobody's talking about this, unfortunately. I'd love to know what is really going on here. How are they different? And that leads me to the next question. How are these yeast genetically unique? And how does this actually equal differences in flavor profiles? And one of the interesting things as far as data is concerned about the analysis from Galone is they identified almost 2.5 million single nucleotide changes, single changes in the genetic code within those 157 strains. And you're like, okay, well, that's not too bad. It's a lot. And considering that the yeast genome is only 14.5 million base pairs in length, that is a lot of change due to these specialized fermentation practices. Now, it's going to be a combination of the practice is going to help guide the evolution, and the evolution then helps guide the fermentation practice. Now, not any one strain is going to have all 2.5 million changes in it, but given the diversity that we see within a simple organism like yeast, it's really impressive that this amount of evolution or change has occurred due to brewing practices. Although we know that there are changes in these yeast genomes, it's surprising how little we know about how those changes are actually linked to specific flavor and performance changes. Galone did a little bit of analysis on this, but didn't go into a lot of detail, and again, hid some information from us that would have been really helpful. So very little or limited information on how specific genes are linked to flavor profiles. So this is where I come in. This is where I really started to get fascinated with this process. So about 4 years ago, we sent a bunch of brewing strains for something called Oxford Nanopore sequencing, or ONT sequencing. We initially started with 5 strains, but we got data back on 4. I don't want to scare you with this, but this representation here, these purple bars, represent a yeast chromosome. So there's 16 chromosomes here. They're all laid out just to show you the length of these chromosomes. In this red bar here, at the end of chromosome 3, I have this region highlighted. And you can see in the red and blue boxes, these represent genes, and they're just genes facing different directions. When we look at the end of chromosome 3, we have an English ale here, a lager strain here, a Norwegian Kvike strain. This is a Norwegian farmhouse ale yeast here, and then another English ale here. When you look at the Kvike strain, it has a chunk of chromosome at the end of the 3rd chromosome that is still present, that has been lost in the other 3 strains. So this is interesting that the Kvike strain is maintaining a piece of DNA that is missing from the other strains. And this contains an alcohol dehydrogenase gene for fatty acids, as well as another alcohol dehydrogenase. These are involved, this one in particular may be involved with removing off flavors in beer. Which is pretty interesting. So this is just not even using any high-powered equipment. This was just a visual analysis. And we find the end of chromosome 3 is present in the Kvike strains, while it's missing from others. Looking at other regions of the genome, again, this Kvike strain is actually missing a chunk of chromosome at the end of chromosome 11 that the other 3 have maintained. And this encodes something called the FLO10, a flocculation gene. This gene is involved with when yeast get stressed out and they get tired, they kind of stick together and then they fall out of solution. And this generally happens when beer production is finished. The Kvike strains tend to stay in solution a little bit longer. So again, another interesting change in this Kvike ale strain that's different from the other 3. And the last example I'll show you is this PAU19 that is present at the end of, I do believe this is chromosome 13. And this gene is involved with mediating stress that's missing from the other 3 strains, but is present here. Now, why this is interesting and why I'm pointing this out is that these Kvike ale strains are traditionally, you find them in Norway. And the methods for brewing in Norway are to use a very warm starting material called wort. This is the soup with the barley that's been steeped in there. It's a tea, literally a tea, with the nutrients in there. Now, what the Norwegians have been doing for hundreds of years, instead of letting that cool down to room temperatures before adding the yeast, they added the yeast warm while the wort was still on the warm side. And what this does is it forces the yeast to evolve for heat tolerance. So what we see here in this wooden ring is I can't remember what it's called, but it is a traditional ring that you find hanging in Norwegian farmhouses, and the yeast is stuck to the surface of this. So if you want to make a beer, you just take this ring and you dip it into the wort, and it inoculates the culture, inoculates your beer. Then you take it out and you hang it on the wall and you just leave it sitting there. So this is pretty cool stuff that this has been going on for centuries. But since this was done warm, the yeast had to maintain a tolerance for that heat so they didn't die. And when you do this with the Kvike strains, let's say you want to make a beer at 28 degrees Celsius. If you used any of the other three strains I talked about, it will make rocket fuel. It will make a beer that tastes absolutely terrible, a lot of off flavors. But the Kvike strains, because of these genetic changes, will make fruity pineapple citrus flavors. Again, citrus coming from Norway seems like an odd idea, but these Kvike strains have evolved to tolerate that heat, and they make fantastic beers at high temperatures. They're really robust yeast. And now the question is, do the extra arms or missing pieces of chromosomes actually contribute to that? So we're investigating that right now. We have a couple of draft genomes, and we should be publishing some data fairly quick on that. All right. So this leads us to the future of the brewing industry. I think with the technology that's been developed over hundreds of years of trial and error, the technology of vessels and the brewing practices have been honed very, very well. So for me, the future of the brewing industry is actually in precision fermentation, taking this knowledge of genetics from the yeast genomes and identifying how changes impact not only performance, but flavor profiles. So just out of curiosity, I whacked in the term precision fermentation into an AI on Google, and I got the phrase, and I'll just read this out: technology that uses microorganisms to produce food ingredients like proteins, fats, vitamins, enzymes, and more. These organisms are genetically modified to produce specific ingredients and are sometimes called cell factories. I agree with most of this definition, but I think for fermentation, it must be highlighted that this is an anaerobic process if we're talking about fermentation. If you're doing it in the presence of oxygen, it's something else. So the definition is lacking something, but the idea is there that we can use these microorganisms, including yeast. We can tune them genetically to start doing the things we want them to do. So for me, this is the last real frontier for the brewing industry, is to develop these new strains through either natural evolution, selecting strains, or genetic modification to get the products we want. Certainly for the pharmaceutical industry, a lot of the cannabinoids have been put into the pathways have been put into yeast. This is something that's being done. We can make polymers with our microorganisms, but there's certainly impacts on human and animal nutrition in various fronts here. I don't want to get into that too far, but certainly this is a huge frontier for the future of not only the brewing industry, but biotechnology on a whole. So I do want to show you this diagram here of how some of these flavor profiles are actually made. The message I want you to take away from this is it's complicated, right? As with anything biological, it's complicated. And when we talk about humans, we talk about plants, we talk about animals, and we try to work out what's actually going on. We're struggling enough with a yeast genome that is only 14.5 million base pairs in length. We're struggling to link that information with flavor compounds on the other side. So I want to run you through a few examples here of how biotechnology and how this has contributed to the brewing industry in the last couple of years. So again, our major issue is that we don't have a good understanding of how specific genes are linked with specific volatile or flavor compounds. How can we begin to look at this? I'll just talk about some of these methods briefly. We can certainly look at evolution and basically putting pressure on yeast over long periods of time to select for the traits we want. This is a long-term goal. Again, we've been brewing for possibly 12,000 years. We do have some evolution, but we haven't linked all those changes with specific changes in behaviors or phenotypes. Natural selection, we can hit yeast with harmful compounds that cause DNA damage, and then again select for the phenotypes that we want. Interspecies hybridization, taking two different types of yeast and forcing them to mate so you get this genetic diversity in the background. It's an interesting concept, but both trying to mutate genomes and forcing these hybridizations is also going to take a long period of time. Certainly not as long as evolution, but this is a very technically demanding, these methods are technically demanding, and it's going to take a while. With our recent developments in molecular biology and genetics, we can take segments of DNA from other organisms and we can randomly insert them into the yeast genome. It's relatively fast, but again, you have to screen for phenotypes that you want. You may not get your target DNA in the region of the genome you want. This can lead to complications. We can use things like homologous recombination, again, for you science nerds out there that know your genetics. Basically, you get double-stranded DNA into a cell that has a region that is very similar to an endogenous region of the yeast genome. The cells try to repair the damage of this foreign DNA, and they integrate it into the yeast genome. It's fast, but it can be inefficient. If we were talking about mammalian cells, this is not something we can do readily. It's easier with yeast, but again, it's a little bit time consuming and technically demanding, but certainly better than the other methods. The last method, of course, is our CRISPR-Cas9, our genome editing technology. This is an extremely fast technology that gives you very efficient integrations at sites of your choosing. And for those of you who are not familiar with the CRISPR technology, I won't go through what the acronym stands for. It won't mean anything to you, but it's basically what this is an enzyme that we give the yeast. And this enzyme is designed to cut DNA. That's all it does, is cut DNA. But it doesn't have a targeting mechanism. It doesn't have a way to decide where it's going to cut. What we do with this enzyme is we give it a guide, a single-stranded guide RNA. And if we make this guide RNA very similar to our target region, what happens is this Cas9 protein, this CRISPR protein, will bind the RNA using this little blue piece of RNA to hold on to, and then it'll find its target inside the yeast genome using this RNA as its guide. Once it does this, it'll make a cut. It'll make a cut in the DNA and create a break. Now, cells, because they don't like DNA breaks, they will try to repair this. Again, if we give it a donor piece of DNA, so homologous to the sequence we want, but has a little insertion, the cell will take over and do its job. It'll try to repair that damage, and it'll insert this targeted DNA into the location you want. I still wouldn't call it very efficient. It's about 0.1%, but it's far better than any other method that we have. It's extremely well targeted, and it works fantastically well. As far as, again, comparative to the other technologies we have. So CRISPR is a really effective tool, and over the last 15 years, we've done a lot with CRISPR. I won't talk about that here, but there's a lot of really cool things we can do with CRISPR. So let's talk about banana flavor in beer. And this comes back to something I said earlier on about modern brewing technologies and how the size and shape of a vessel can influence the fermentation process. So using modern brewing practices, we tend to lose banana flavor that the yeast are producing with our modern setups. And what we have here, I'll just take you through what bananas actually The banana flavor actually is its isoamyl acetate. It's this molecule here. So what happens during metabolism and yeast growth? They'll produce this isoamyl acetate. And then two enzymes in the yeast, this alcohol acetyltransferase 1 and 2, they're responsible for taking this acetyl-CoA group and attaching it to this alcohol group here to produce isoamyl acetate. And this will give you pineapple and banana flavors. These two enzymes, ATF1 and 2, are responsible for making a bunch of different flavors, taking these alcohols and turning them into acetate esters. You get a floral flavor with this 2-phenylethyl acetate. Isobutyl acetate is just a general fruity flavor. Ethyl acetate isn't a good one. This gives you kind of a solventy flavor. But these two enzymes are really important for that banana flavor. Now, coming back to the point I just made about modern brewing techniques, traditional brewing was done in large open vats that were very wide and very flat. So there isn't a lot of pressure of gases building up with the fermentation. So the fermentation releases CO2. But also because these were wider and shorter, there wasn't a lot of liquid pressure on top. With modern brewing techniques, we have these very, the width of them isn't very great, but the height is. When we have this height, it actually puts pressure on the yeast that is down below. And this suppresses the expression of ATF1 and 2, surprisingly enough. Even though here we have higher risks of contamination, these are much better. I think these are better fermenters overall as far as risk and what we can do with them. But it has caused a change in the yeast because of this different geometry. It's where a systems biology comes back in. So by doing our fermentation in these types of vessels, we suppress ATF1 and 2 just by putting this natural bit of pressure on them. So what this group did in 2022 was they did a genetic screen of yeast to find out what had happened over the evolution using these high stacked up vessels. And they found that this gene, MDS3, was mutated and suppressing the banana flavor. Now, this isn't a mutation in ATF1 and 2, but because of the mutation in this gene, ATF1 and 2 don't function as well as they used to. So this gene, when you look at its function inside the cell, it says it's a regulator of pH-dependent morphogenesis. Not really sure what that means either, but it seems really far unrelated to the production of isoamyl acetate. What we do know from this is that MDS3 is a part of something called the target rapamycin pathway. This is one of the major energy sensors inside of the cell. It's involved with nutrient sensing and stress sensing overall. But it must be an indirect interaction. When these authors did some genome editing and repaired this gene so it was functioning again, they increased the levels of isoamyl acetate back up to 145% of what it was in the mutant. So given the link between energy sensing and acetyl-CoA, again, that molecule that's modifying isoamyl alcohol, this is a readout of cellular energy. So there could be a tie in there. It's just really indirect. So I should have, pardon me. Okay, yeah. So they were able to increase this banana flavor and actually add it back to their beers. So that's a pretty cool application. Another application of CRISPR has been in something called phenolic off flavors. And these are clove-like, peppery, spicy flavors that you get in some beers, especially your Belgian ales. I love Belgian ales. I love this flavor. But it's considered an off flavor in lagers and other ales like English ales. And what this clove flavor is this 4VG. And it is a result of this ferulic acid being converted being decarboxylated by FDC1 to generate that 4DC. So what the authors of this paper did was they used CRISPR to modify FDC1 to turn it from a phenolic off flavor positive strain to a phenolic off flavor negative gene. And again, using CRISPR, They did this targeting, and all they did was they changed the sequence of F81 or FDC1, and they engineered a single base pair change to generate a stop codon. And for those of you who are familiar with your molecular biology, again, this one nucleotide stops the cellular machinery from making this enzyme by telling the translational machinery to stop making the protein. So very simple, very easy to do. And again, with the CRISPR technology, worked really well. And just from this bar graph, they did this in a bunch of different strains. Again, this is the Verstreppen group, same one as the one from 2016, where they did this. And you can see that these are the normal levels of ferulic acid that were present. This is at the start, and they maintain those levels so you weren't producing this for VG. So really cool application of this. But again, if you don't know which genes are responsible for making which flavors, you can't do this. It's really, really challenging. You need that genetics knowledge. So definitely the future of brewing will be to link the genetics with the flavor compounds and then start doing these modifications. And we're starting to do this, but we just need a little bit more background. We need more knowledge in what we call the functional genomics. How does a change in a genome equal a change in function? And of course, with this, we're specifically focusing on flavor compounds. Certainly, we could talk about performance and other applications, but yeah, for the craft brewers, it's the flavor compounds. So coming back to the brewing industry and the current challenges that might be faced with brewers in the next little while, certainly market saturation. There has been an explosion in the number of craft breweries, and I think that is hitting a mark where, yeah, where it's tough to maintain that. So market saturation, but not only that, product innovation. How do you differentiate yourself as a brewery from all the other breweries doing really cool things out there? Again, I love most breweries, but to select out my favorite one for doing something truly innovative, it's a challenge because there's limits on it right now with the yeast that's available and the ingredients that are available. Unfortunately, with some of the breweries that are not doing well, they potentially give the market a bit of a black mark, a little bit of a black eye because of poor practices and poor yeast performance. If you make a beer that isn't good and you're just starting to get into craft brewing, that can turn you off. With the breweries themselves, they make excellent recipes and they make excellent products, but they may not be able to take care of their yeast. So the yeast performance could be a problem there that needs to be dealt with. Certainly health trends are a consideration here as well. I don't know how many of you out there like these low-calorie beverages, the seltzers, the teas, and the lemonades. I personally am not a fan of soda water, so I'm not a big fan of any of these, but certainly with health concerns and people watching their caloric intake, this is a trend that's upticking and taking away from the craft brewing industry. It's important to note here that a lot of these beverages, the vast majority of them are not fermented, they're blended. They make a juice or a mixture and then they add in ethanol. I think Rebellion Brewing out of Regina is one of the very few that actually has a fermented tea that is those I like because it's an actual tea and it's an actual fermentation. It isn't a blend, but certainly the revenue around this market is fairly significant, about $34 million US in 2024 for Canada. Sorry about the US dollars there, I didn't do the conversion. Another trend, though, for the brewing industry is certainly the demand for low alcohol containing beers. People still want to drink a beer; they love the taste, but they're trying to avoid the alcohol. And these are certainly becoming prevalent in the market. The technologies have become much better than what they used to be, and what we're defining as a low alcohol beverage is anything below 0.5% alcohol per volume. Now, I've been doing some investigation myself on how breweries are actually producing these beers. One of the ways that could be used is by heating up the beer after it's done to boil off the ethanol. not really boil it off, but to evaporate it off and reduce the ethanol content. Unfortunately, the volatile compounds tend to evaporate right around these times too, so you can lose a lot of that flavor. But I know it's probably the method that most of the larger breweries are doing, probably a distillation method to recapture those volatiles and re-add them back to the beer. But I'm really not familiar with the chemistry behind that. So that's one method of doing it. The other method is to prevent ethanol from actually being made in the first place. I just have a couple of Canadian examples here of beers that actually aren't bad that they're alcohol free. I generally find that their taste is a little bit, it's not a beer for me, right? I drink beer not to get drunk. I love the taste of beer, but I just find that there are certain things missing from these alcohol-free beers. There's a mouth feel that's missing from them as well that tends to be a little off-putting for me. But again, that's my personal choice. So the second step, prevent ethanol from actually being made. Again, using genetic technologies, we can alter or select yeast that have the inability to utilize maltose. Maltose is the sugar that we find in wort and beer from barley. It is a primary energy or sugar source from the wort in the beer. And what the yeast do is they utilize that and they convert it into ethanol. So that primary sugar that's being fermented. However, maltose doesn't transport across cell membranes and into the yeast readily. It has to be actively transported. So if you don't have an active maltose transporter, you do not undergo very much fermentation, and that sugar stays in solution. And again, for you genomics geeks out there, in chromosome 2 of yeast strains, you find this cluster of maltose related genes. Mal31 is the transporter. Mal32 is the maltase that breaks this maltose molecule into smaller sugar units. And then this Mal33 is an activator of these genes. So what you can do is you can look for mutations or generate mutations in the Mal31 gene or genes if there's multiple copies of it using CRISPR or that forced evolution and screen for your phenotypes. So this is a great way, but you end up with a very sugary, watery solution with not a lot of the volatile flavors produced in that. And there are concerns with this because there is so much sugar left over. If you have a few microbes like lactobacillus fructivorans laying around, they're going to pop right out of solution, and you're going to end up with not only contamination, but what we call secondary fermentations, where the bacteria start to produce ethanol, and it's now no longer an ethanol or a low alcohol beverage. If you do this after packaging and there's a little bit of bacteria there, the cans start to explode because they're fermenting in the can, and there's a lot of this sugar left over. So it is a major concern and a major challenge for brewers moving forward is how do you get these low alcohol beers using yeast but without creating all these adverse conditions. So I'll stop there. Just some concluding remarks. Again, beer and our fermented beverages have been really important for civilization and our development. There's certainly a huge part of the Canadian economy. The impacts are probably far more reaching than most people realize. And the next frontier will be the genetics and genomics for that precision fermentation that really provides brewers with an advantage. And then we have to look at the health trends and what consumers actually want. But tying those things together, the economy, our knowledge of genomics and biotechnology with what consumers are demanding, yeah, that'll be the move forward in the next 5 to 10 years. So with that, in conclusion, I'd like to thank my co-founder and Jordan from IntelliYeast Laboratories and Fina Nelson, who's a PhD student in my lab, who's working on an ADF-funded project dealing with linking genetics with flavor profiles. So with that, I'd like to thank the Food Center for the invitation, AgWest Bio, Innovation Saskatchewan, and Opus for the help with IntelliYeast, and the government of Saskatchewan, the ADF program, NSERC, and Proxima for their assistance with the research side of things. So thank you for your time, everybody. Really appreciate it.
Thanks so much, Chris. Really appreciate all this great presentation, webinar, and also your insights. There will be several questions, I assume, but I just like to start primarily on this whole regulatory path of the genomics, the new yeast strains developed within this whole business. Like how easy to get them approved, or what's the process of those products to be utilized in the market? Can you please start elaborating that, please?
Yeah, that's an interesting question, and I've been doing some digging, but there are no clear answers, of course, because the yeast is being potentially filtered out at the end of the brewing process. We can modify those yeast, and we don't have any regulations around that.
Mhm.
As far as I know. And again, we're just for myself, I'm just in the infancy of starting to work on this. But there are certainly strains of yeast that are available commercially that we know are genetically modified. They're posted as being genetically modified, and it's fine, right? So as far as the requirements to tell people that it's been genetically modified, I'm not sure there is any requirement at this moment.
Okay.
So
Thank
now my insurance,
you.
if I used a genetically modified organism in my lab, I definitely have to let my insurance company know, right?
Yeah.
There are different regulations there from the commercial side.
Yeah.
Yeah.
So in terms of the commercialization timeline for these new yeast strains, how long does it take to develop, like from your process, developing and establishing and to the sales? Like how long does it take? Can you talk about that a little bit?
I would say with a new strain that has been genetically modified, if we already have a gene identified that we want to target, 6 months to a year, and it could be out in the market. It doesn't
Commercial.
take very long at all. A few trial fermentations to make sure the yeast is doing what it's supposed to in the beer.
Mhm.
Yeah, not long at all.
That's great. And in terms of the whole growth in the non-alcoholic beer, I mean, the overall, like what we know as well, is basically the evaporation or the boiling process, which is like a fairly energy cons like a consuming process that we use quite a bit of energy and steam and heat and so forth. Like that whole the new development, and there's definitely more interest in the market with the non-alcoholic beer and similar products, fermented products. With this type of new yeast strains, are there anything available? Is this something that you are working on? Like, what is the pipeline looking at, or are there any things out there in the market? Like, can you talk about that?
Yeah, there are definitely in the market yeast strains that have maltose transporter mutations. And I've been involved heavily with the homebrewing community. There's a lot of people using them and a lot of subpar products being made. Even though this is the methodology currently, I don't think it's a very effective. Uh, I don't think these yeast strains are actually very effective, but they are commercially available through almost every major yeast company.
Company.
But
Okay.
it's one of those things that it'll get better with time as we gain a little bit more knowledge. We'll improve on this technology, and I'll admit I'm working on this myself, taking a different approach though. I
Okay.
won't reveal too much here, but certainly, yeah, this is a major focus of biotech right now is to get this done for the market and for
That's
the
great. Because I mean, that's definitely a growing field. So
for
we
sure.
have a question from Henry Ha. He's basically asking, how do you see the future microbreweries getting tailored strains from specialized labs? What would that be a way for them to distinguish themselves from competition? Is it feasible?
Absolutely. I think specialized labs, I think Berkeley Yeast out of Berkeley, California, they do pretty much nothing but genetically modified yeast that is tailored to making specific flavors. Now, any one brewery getting their hands on a strain exclusively, I don't think that would be in a company's best interest to sell to only one brewery. One way a brewery could really distinguish themselves is to go into their local environment and isolate yeast from that environment that are turn out to be good for brewing, that give that really local flavor. For example, one of the long-winded answer, I apologize, but one of the things we were doing in my lab is actually going out into our local environment here in Saskatchewan and finding yeast strains and doing test fermentations. And this has actually been successful in finding very unique strains that then you can catalog for yourself. And then that brewery would make a very unique beer with that strain and not give it out. So I'd say that would be more the way a brewery could really distinguish themselves and differentiate themselves, but it's a lot of legwork. So
Great.
yeah.
And in terms of the yeast genomics that what you are working and industry working, are these typically specific for barley, but like there's also other carbohydrates in the beer like wheat and lentils, oats, rice, sorghum, and other products globally. Are they working similarly when there's maltose in the process, or like do they have different interaction based on the raw material in terms of flavor and so forth?
Absolutely, they'll have different performance. Most of the beer strains are programmed over evolution. They're specifically geared for maltose. So you can make some, for example, rice lagers, but they still have to have barley in them because they're not using the same carbohydrate sources. Certainly, wine is an excellent example of strains that are tailored for sucrose and glucose fermentation. If you give them any sort of maltose, they don't like it at all, right? They do a really poor job with their fermentation. So evolution is taking care of some of that. But if you're starting to use other sources of carbohydrates, you really need to pay attention to what strain you're using and the genetics behind it. Right. Just as another example of that, most of the ale strains use maltose and maltotriose. So no more than 3 glucose molecules hooked together. If they're any longer than that, that carbohydrate stays in solution and the yeast cannot utilize it. So having ways to break that down and make it metabolizable, yeah, for alternative carbohydrate sources, yeah, you really have to pay attention to what types of molecules are there.
Great. So the last one I like to just to wrap up, and again, thank you so much. Can we preserve the IP on some of these new developed yeast? What's the overall IP landscape like for each? Like, are we talking about more like a platform, or is it more like each species, each strain can be like IP'd and patented, and how we can basically add those in the Canadian IP landscape?
Yeah, definitely there is a track record of companies developing yeast strains and patenting them. There's a couple of Omega labs in the States that if you are caught using those, you are going to pay some fines. You do need to have those licensed out. Most of the strains are not, but there's certainly IP avenues for that. You just have to find a way of tagging that strain and making sure that it does not escape into the market. And an example of this is that if you make a beer with the yeast that I have developed and you do not filter that beer, somebody goes out, buys the beer, and it has the yeast in it, you can then grow it on your own. It escapes the IP route. So if you are producing something that you do not want it to escape into the general public, you need to filter out every yeast cell or make sure that it's pretty much dead before you package that product. It's complicated. I'm still doing a lot of digging around here. It is a new frontier for IP and legal issues. So yeah, something I'm certainly interested in, but yeah.
Thank you. Great. Thank you so much again for this wonderful webinar and your insights. I'd like to thank our audience for joining us. Thank you, and wishing you all a great day.
All the best, everybody. Thank you very much.
Thank you.